Okay, so welcome to all. My name is Mathieu Canel-Valliere. I am a postdoc with Joseph Brasch and Kristen Lynch at the University of Pennsylvania. Very happy to be here to host the very first journal club of the OI-RNA COSI. Before we get started with the journal club, I want to take 1 minute to introduce what the OI-RNA COSI is. So it started as a community of special interests in the context of the ISMB meeting. So basically every year, a group of integrative RNA biologists gather at this meeting. And what we want to do now is to take this group and make it into an online community so that we can interact with more people throughout the year. We already have a website, irbgroup.org, that you can use to see news, events that we announce there, information about the ISMB meeting as well. We now have a Twitter account that's getting more and more active at iRNA COSI, and this is one way that the community can get to interact with others. I would encourage you to follow at iRNA COSI. And also mention this handle if you want us to retweet information that you might post about upcoming publications that you have, preprints, if you're putting together a meeting or any kind of event that you think could be of interest to people doing integrative RNA biology. I would encourage you to get in touch. If you're wondering what we mean by integrative RNA biology, I will tell you that if you do anything that's RNA research and you use computers, you are likely an integrative RNA biologist according to our definition. If you develop high throughput technologies, new techniques to study RNA, you are likely an integrative RNA biology. So we want you to become part of this community, give us feedback. We have an email, oirna@irnacosi.org, that you can use if you have ideas about activities that the community should be putting together. If you want to contribute as well, please reach out. So today we have a journal club that's part of this community. It's one of the very first events that we're putting together. We have other projects on the way that we will be announcing in the next few weeks and months. Today's journal club is supported by the International Society for Computational Biology and the RNA Society, and I want to thank Diane Kovacs and Seth Mulholland of ISCB, as well as Christian Baker of the RNA Society, for helping us organize and advertise the journal club this morning. Things have been pretty smooth so far in organizing this journal club, so much so that we can already announce the next journal club. So next month, we will have Anna Fishbein, who is a postdoc in Chris Burge's group, who will be with us to talk about dynamic determinants of co-transcriptional gene regulation. That will be related to their recent report about the discovery of exon-mediated activation of transcription starts. So that will be on April 21st at 11 a.m. Eastern Daylight Saving Time. So it's also on a Tuesday, same time as today. So indeed, we are very lucky to have two very strong speakers and two very interesting papers to launch our journal club with Anna next month. And with today, this morning, Furqan Fazal, who is joining us. Furqan, good morning and welcome.
Hello, hi. Thank you everybody for tuning in, yeah.
Yeah, well, thank you very much for agreeing very kindly to be our first speaker. It's a big leap of faith of you to accept, not knowing how this is going to go, how many people would attend. So I thank you for being with us. So just to put some context to this journal club, the way we want it to be is as close as possible as we would see it in a regular journal club. So if we were all gathered together in a room, so we will try to take questions on the fly. You will see at the bottom of your screen, there is a Q&A icon. If you hit that icon, you can ask questions. The questions that you post there, I will be the only one seeing them. I will be the one who will be taking questions for Furqan and asking them as we go during the presentation. We will also have time for questions at the end of the presentation, but please feel free to type your question during the presentation. Furqan, I might ask you to pause every now and then between slides to see if we have questions coming in. One thing that I might suggest that you do, and I'm talking to the attendees now, if you know that you're going to have a question that's going to take more than just 2 seconds to type, maybe just type question or I have a question so that I know that you have a question coming so that we can pause to give you some time to get your question in. So this morning, Furqan, you are a postdoc in Howard Cheng's group, and you are here to discuss your recent report about APEX-Seq and how you've used the technique to identify a number of RNA transcripts in different cell compartments. This is a technique that you developed in collaboration with Shuo Han from Alice Ting's group, also at Stanford University. And since we have the first author with us today, we should be able to answer all of the questions because you know everything about this. Is that correct, Furqan?
Yeah. Yes.
Yeah. So this is amazing, and this is the idea behind the Journal Club. So instead of being left hanging with unanswered questions, we will have the answer to all of those questions today. So I'm very much looking forward to that. And Furqan, I will let you take it away. I will intervene if we have questions coming in. So good luck, and I'm hoping that this will be fun, and I think that should be fun.
Okay, great. Let me launch the slide. Let's see. Okay, so you can all see the slides.
I can see it all right. It should be
okay, great.
Should be all fine.
Okay, great. Again, thank you so much for having me as the inaugural speaker. So I'll give this journal club on, again, a recent paper from the middle of last year, APEX-Seq, looking at subcellular RNA localization and doing this transcriptome-wide, that is for thousands of transcripts simultaneously. And so the talk will be divided into 3 sections. And I know this is following a journal club, but I also want to make it so that in the interest of time, I cover some of the more interesting, more widely applicable panels in the figure, but feel free to ask me about other things that I have not had a chance to cover. And so again, I'll divide this into 3 parts. Really, this beginning will be an introduction, where I introduce the area of RNA localization, tell you a little bit about the existing techniques out there, and then introduce APEX-Seq and some validation. And this is just to lay the groundwork of this technique and the context in which it was developed. And so you could probably ask me questions at the end of that part. Then the second part will be really looking at the transcriptome maps that we've generated using APEX-Seq and how that correlates with other biomolecules such as proteins, as well as DNA and the genome architecture. And the last part, I'm going to focus on one particular location, the outer mitochondrial membrane and the mitochondria in general, and how using APEX-Seq we can understand some of the principles that underlie RNA localization, really emphasizing why this is an interesting and important area of research. So, as kind of all of you know, that a cell is a highly organized entity. And so this includes both prokaryotic cells as well as eukaryotic cells. And within a cell, location informs function. So in a eukaryotic cell, there's a nucleus that's associated with transcription and splicing. The cytosol is where a lot of translation happens. Secretory pathways go through, for example, the ER membrane, the endoplasmic reticulum membrane. The mitochondria is associated with respiration. And so we're familiar with the idea that specific parts of the cell are associated with specific functions. And moreover, knowing the location of the biomolecules within cells, so proteins, as well as small molecules, and even RNA, kind of informs what their functions are likely to be. And again, this is true for proteins. So many of you know of proteins that localize to specific parts of the cell, where they're engaged in specific functions, but this is also true of RNA. And this includes both coding RNAs, as well as the entire plethora of non-coding RNAs, long non-coding RNAs, smaller RNAs, and so on. And so in general, from decades of studies, we know many RNAs are spatially localized and locally translated. And there's beautiful work in many different model organisms going back to over 30 years now. Some of the seminal work was done, for example, in Drosophila development. And so in Drosophila early embryo, many RNAs are known to localize, not just expressed by specific cells and by specific cell types, but also within cells, they localize to specific parts of the cell. So for example, the nucleus or the cytosol. And so this is images showing the location of different RNAs, where the RNAs are shown in green. And you can see, so this is visualized using fish fluorescent in situ hybridization, where a fluorescent oligo is used against the RNA of interest, and the nuclei are stained in red, and you can again see different patterns for different RNAs that are important in development. RNA localization, RNA subcellular localization is also important in mature cells and adult cells, and it also can be dynamically regulated by the cells and by organisms. So for example, in these intestinal epithelial cells, they actually polarize the RNA contents in response to food gradients. So food is on one side of the cell, and the cell responds by essentially polarizing its contents so that it can more efficiently utilize the resources available by having a lot of local translation of messenger RNAs near the food source. And so in general, it's known spatial localization influences many aspects of gene regulation, including RNA folded, editing, splicing, translation, degradation, even the fate of the protein that's encoded. And a cell invests a lot of resources in making sure RNAs end up in the right place at the right time. So this includes the well-studied process of nuclear export, where many RNAs, typically messenger RNAs, are exported from the nucleus to the cytosol, where they're translated. Within the cytosol, they may be moved by cytoskeletal motors from one part of the cell to the other. There's also degradation machinery to degrade RNAs in specific parts of the cell. And so although we kind of know of many of processes involved in really localizing RNAs, at this point, we still don't really appreciate fully the scope of this process, the extent, and really what its importance is. And part of the challenge really is that there haven't been very good techniques to look at RNA localization transcriptome-wide, that is for hundreds, thousands of RNAs simultaneously. So if you're interested in a particular RNA or RNA coding for a protein or a long non-coding RNA or another type of RNA, you can use techniques such as live cell imaging or FISH, fluorescent in situ hybridization, to kind of see where that RNA localized within cells and even perhaps study its dynamics in living cells. But if you want to do this process transcriptome-wide, that is for thousands of RNAs simultaneously, that continues to be challenging. So fortunately, there have been some techniques and there has been progress in this area. So one technique is, of course, fractionation. So this can include biochemical or physical fractionation, where RNAs, where you fractionate a cell, let's say into a nucleus or in a cytosol or some different location, and you interrogate the RNAs in those locations using sequencing. And so although this has been very useful in some locations, such as nucleus and cytosol, for many interesting locations within cells, particularly membrane-bound and membraneless organelles, they're difficult, if not impossible, to access using fractionation. Furthermore, fractionation can introduce impurities during the purification steps. And so really in the last 5-6 years, there have been new techniques to try to address some of these challenges. So some of them are imaging-based, such as MERFISH and SeqFISH, and where RNA probes are used sequentially to look at the location of not just one, but tens, hundreds of RNA molecules. These are very powerful techniques that are still being developed. Some of the challenges are these are done on fixed cells. They require prior knowledge of the RNAs of interest because you design probes against those RNAs. And so it's challenging to really study different isoforms and really the complexity of the transcriptome. And also there might be limits to how many RNAs you can study because you need to put all of these fluorophores within cells. There are some sequencing-based approaches such as ribosome profiling, where you can access the RNAs bound to ribosomes and sequence them, and that gives you the RNAs in some locations, but this is of course not a generalizable technique. And so really in my postdoc, in Howard's lab in collaboration with Alice Ting's group, we worked on developing a new technique to essentially look at RNA subcellular localization in a scalable, affordable, high throughput manner, and use that to build a preliminary subcellular atlas of RNA localization. And so the way this works is we leverage an enzyme called APEX. So APEX is an engineered ascorbic peroxidase, which was engineered in Alice Ting's group. And APEX-Seq involves live cell proximity biodylation of the RNAs by this peroxidase. So the way this works is we express APEX as a fusion protein where it is fused to a protein or peptide that directs APEX to a particular location within cells. So for example, many of you are familiar with the NLS sequence, the nuclear localization sequence. So if you express APEX as APEX NLS, then it will go to the nucleus and allow you to get the transcriptome of the nucleus. In this example here that I'm showing, APEX is localizing to the outer surface of the mitochondria because it's fused to a protein MABs that goes there. Then we activate a reaction, which is a 1-minute labeling, providing biotenphenol and hydrogen peroxide, and it results in the RNAs in close proximity being biodelated. And then what we do is we stop that reaction, we quench it, and at that point, the RNA localization is essentially registered, and then we enrich for those RNAs that were present at that location using streptavidin biotin chemistry because they're labeled with a biotin tag, and then we sequence them. And so this was, again, this is the journal club that I'm covering. And the key insight we had is that APEX was actually developed for subcellular proteomics, where it is shown to have high spatial and temporal resolution. And what we reason is that the radicals that are produced during that reaction, they're promiscuous. So they will label proteins and allow you to get subcellular proteomics. But what we also found is that it will label other kinds of things. So it should label RNA, small molecules, even DNA. And so during my postdoc, I worked on extracting these RNAs and sequencing them and really showing that this technique really worked. And I should mention that 2 other groups have also shown that this technique can indeed be used to directly label RNAs. And so I'm not going to focus much for the purpose of this particular audience and talk on showing you a lot of the gels and the evidence for the direct labeling. But what I'll just summarize and say is that it seems that the radicals preferentially label G-rich regions of the RNA. And this technique does indeed work also in a sequencing-based approach. And so when we target APEX to the inside of the mitochondria, we recover all of the RNAs present at that location. So recall that the mitochondria has its own genome. It produces its own RNAs. So it produces 13 mRNAs and 2 ribosomal RNAs. And so when we perform the APEX-Seq experiment, where we get this transcriptome-wide information, along the y-axis is the expression level in unperturbed cells, expression level in the APEX-Seq experiment, and along the x-axis is a control where we perform everything else identically, but we omit one of the reagents, hydrogen peroxide, required for the labeling. And so you can see that when we perform this sequencing-based approach, so it's kind of like an RNA-seq experiment, but using APEX, what we see is most of the transcripts lie along the x equals y axis, so they're unchanged in the 2 conditions. But what we see is robust enrichment of all the 15 mRNAs and ribosomal RNAs in the mitochondria.
Yeah, Furqan, so I'm coming back online because we have a couple of questions. The first one would be, so because the biotin phenolic C radicals are so promiscuous, did you run into problems with the purity of your pull downs, and what are the kind of wash steps that you had to go through to get a cleaner RNA fraction at the end of it?
Yeah, so maybe I can just quickly, let's see, show a different slide. Can everyone see that slide?
Yes, we can.
Okay, so so the idea is that yeah, so it does label different kinds of products, but we've confirmed multiple times that we're actually just extracting the RNA. So we're not looking in these experiments at proteins or different molecules. When we use an RNase digestion, the signal disappears. So this is this is a streptavidin biotin dot plot where we're labeling the RNA within cells, and then we're extracting it and running it on a dot plot to see the signal from the biotin phenol. So this was in the original paper. We have even better data since. And so, that was initially a concern, but we're confident that the signal we're getting is just from RNA.
Okay, I have a few more questions. Alex Price asks, besides G-rich, are there structure determinants that determine whether an RNA can be labeled or not?
Yeah, that's a good question. So that's something we looked at, and actually this is the slide that also shows it. So I also ran a gel, doing this labeling in vitro, using not APEX, but a related molecule HRP, but the chemistry is likely to be very similar. So to see where the RNA is labeled, because it induces stops when you reverse transcribe the RNA to make DNA. So that tells you where the RNA was labeled. And so it's labeled at G-rich regions, and at least for the RNA that I looked at, 5S RNA, it doesn't seem to care what structure it has. So it will label structured as well as unstructured regions. Yeah.
Okay. Well, Jessica Silva Fisher asked if you only detected mRNAs or if you can also detect long non-coding RNAs, but I think this is something that you will get to in a few minutes.
Yes, I will.
Okay. Joseph Brash asks, well, he says that you had a statement about how the method is free of artifacts. That's a pretty strong statement. So can you tell us more about the limitations and the biases of the method?
Oh yeah, so I'm going to tell you some more about that on actually the next slide, telling you a little bit about the limitations that are out there.
Okay.
So yeah.
Okay, so let's move on then.
Okay. Yeah, that's good to go to the great question. Okay, so I showed you data for the mitochondria, which is actually a pretty easy location to get the transcriptome because it is bound by two membranes. So it's kind of difficult. So things remain within the mitochondria. So to really test how well this method works, we actually looked at also the transcriptome of membrane-bound organelle, the ER membrane, which is, this is the cytosol facing size. So transcripts can diffuse in and out. But at the same time, although it's a challenging location, there are existing techniques out there to look at the transcriptome of the ER membrane based on ribosome pull down or ER ribosome profiling that we can use to kind of validate and benchmark APEX-Seq. And so when we perform the APEX-Seq experiment, which I'm showing here, so this is where we target the ER membrane and we recover a transcriptome. Along the x-axis is the enrichment by APEX-Seq, where, so you can see that so on the x-axis is the enrichment. So high values, high positive values suggest that it is strongly enriched by APEX-Seq. And the y-axis is the confidence or p-value in the data. So this is a so-called volcano plot. And as we expect, we recover robust enrichment of RNAs that code for secretory proteins, which is what we would expect. So although this technique is quantitative, for the purpose of this analysis, what I did is make a ROC or receiver operator curve and take all the RNAs above that cutoff and say they were present at the ER membrane. So when we do this experiment, we recover over 1,000 different transcripts at the ER membrane. Over 2/3 of them have been recovered by other techniques, including fractionation and ribosome profiling. So we can ask what are the transcripts that are covered specifically by APEX-Seq, but not by some of the other techniques, and they tend to be lower abundant transcripts. So if you look at the expression level of these transcripts in unperturbed cells, which I'm showing you, what you can see is the ones that we recover uniquely by APEX-Seq tend to be lower abundant. So it's a little more sensitive to lower abundant transcripts. Then we can ask the question of coverage. So of all the transcripts that we know or we expect to be present at the ER membrane, what fraction do we actually recover? And so to do this analysis, what we did is we made a list of 71 transcripts that we expect to see at the ER membrane and see what fraction of those we recovered. And this list was based on doing a literature survey by my colleagues. And so what we see is that 50 of these transcripts, of these 71 transcripts, are recovered by APEX-Seq, which is comparable to existing techniques out there, so ribosome profiling and fractionation. But what's really cool is that unlike some of these other fractionation-based approaches, the same generalizable technique can be used to really target any location within cells, which again includes membrane-bound and membraneless organelles. Furthermore, at least for locations where we had information, such as for nucleus versus cytosol fractionation, the APEX-Seq data was cleaner than fractionation, as it didn't have the mitochondrial contaminants that can be present in the cytosol data sets, as well as ER contaminants present in the nucleus data sets. And so this is really kind of laying the groundwork for what I'm going to talk in the next slides, presenting some of the transcriptome-wide data. So let me stop here. Yeah.
Yeah, Furqan. So regarding the, so you were applying abundance of transcripts that you're picking up here. I have a question from Joseph Yeselman, who asks about the signal to noise ratio. He noticed that you're applying in log2 fold for the enrichment, and that you would probably be picking up 1,000 or 1,000 copies of transcripts, which seems low. So can you tell us about how sensitive the method really is at this point?
So the FPKM is definitely lower than 1,000 to 10,000 copies. I think, let me kind of rephrase. So of course, this is a sequencing-based approach, and so we're limited by how many reads we get. So that would be true of any sequencing-based approach. Right. So in these experiments, we've sequenced around 35 million reads. And so typically, if an RNA is strongly localized, you need about 100 counts to really do the analysis properly using DE-seq or any other method. And if it's not that strongly localized, you may need 1,000 counts. So it is still a lot sensitive than some of the other techniques, but of course, that's a limitation. So yeah, if it's very low abundant, then you just don't have the confidence to call it as enriched.
Right. I have another question here, probably more technical, from Matt BED, who asks about what you think could define which transcripts were identified in the ER fractionation that you did not see in APEX-Seq.
Yeah, so transcripts that do localize, so they might localize to the ER membrane, but also to, let's say, the cytosol. So those might, they are definitely more challenging to recover. So we do recover fewer of those. So the ones we recover kind of specifically go to the ER membrane, but not the cytosol.
Okay.
So yeah.
Yeah, I have another good, interesting question here about the fact that you're using hydrogen peroxide for the labeling. So are you worried that using hydrogen peroxide, and this question comes from Razim Barutku, so are you worried that the hydrogen peroxide could lead to aberrant localization due to reactive oxygen species associated stress, and could lead to mislocalization of the signal that you're picking up?
Yeah, so that was a concern based on the data we have, which let me just quickly show maybe a slide, sorry. So based on the data we have, I don't think that's a concern, but that, that was something we were concerned about. So let me kind of quickly say, so just show this. So actually, yeah, so so on this slide here, if you can see it, so this is comparing what we recover by APEX-Seq compared to fractionation, looking across the transcriptome. So this would be comparing really a fractionation nucleus versus cytosol versus APEX-Seq. And so you can see that really there's very good agreement, not just qualitative, but quantitative agreement. And when we disagree is where there were contaminants in the fractionation data. And so, so really it's a quantitative agreement between them. And also I'll say that in unperturbed, so in all of these experiments, there's a control, and the control is where we omit hydrogen peroxide. And so in that experiment, which doesn't kind of answer it, but I just want to give some context. So when we do that experiment, which is a control, it actually looks like a wholesale RNA-seq experiment. And so this is looking at the APEX-Seq data versus just RNA-seq. And so again, a control kind of looks like an RNA-seq, and then we add on that the hydrogen peroxide, which I'm showing here, which agrees well. So again, that is a concern, and it could be that in some particular locations that might be a challenge, but at least for the ones where we have good data, we seem to have very good agreement, not just qualitatively, but quantitatively.
Right. Okay. Thank you. And one last question before we move on, just to, I guess, to summarize the steps that you follow in the APEX-Seq method. So can you summarize so going from the APEX-Seq to the sequencing, the steps?
Yeah. So first is, of course, making the construct. So there are different approaches. You can use CRISPR. You can use lipofectamine, which is really quick. You can use lentiviral, which is a little slower, but I think that's the way we've done it, and it seems to be pretty robust.
All right, so this is how you localize the expression of APEX to a specific cellular compartment?
Yeah, so how you introduce the plasmid that will express APEX in a particular location. You confirm by imaging that it does indeed localize, and I'm going to show that, just that in the next slide. So once you have it, what you do is you grow the cells up. When you're ready, you incubate it with biotin phenol, and then you add the hydrogen peroxide for 1 minute, and then you stop the reaction using quenching buffer, which we have in the paper, and then you just extract the RNA. So you can use your favorite extraction techniques. So we use the QIAGEN kit, which of course is in very big demand these days, but that's what we use. And the idea was to make it really simple and accessible, but you could use also other things, phenol, chloroform extraction. So once you extract the RNA, then you do an enrichment where you pull down the RNAs using streptavidin biotin pull down. We have a protocol. It takes 5-6 hours. Then you have RNA that is biotinylated, and then you just make libraries out of those.
Okay,
and so, yeah.
Yeah, we have a follow-up question, whether you would be able to use to do the APEX reaction in fixed cells. I would suspect that you might kill the activity of APEX if you do that, and you would do that in order to limit the diffusion of transcripts, I guess.
Yeah, we haven't done it in fixed cells. So APEX has been used for EM imaging. It's a different system, but I expect that it would also work in fixed cells. But extracting the RNA is, of course, one extra step to really extract RNA from fixed cells. So the labeling would, I expect, would work just fine. It's the extraction of the RNA. But yeah, it will work.
Okay, thank you.
Yeah. Okay. So now I'm going to talk a little bit about some of our findings, and let me go to the next slide. Okay, so using APEX, we targeted many different locations, including locations in the nucleus and the cytosol, to really build this subcellular atlas and really understand the extent of RNA localization. And these were done in, again, human cells, so human embryonic kidney cells. And so we confirmed by imaging that the APEX tag did indeed localize to the locations of interest. So these include membrane-bound organelles, such as the outer mitochondrial membrane and the ER membrane, some membraneless organelles like those nucleolus, and kind of dynamic regions such as also the nuclear lamina and the nuclear pore. And I'd like to emphasize that some of these locations, they're quite close to each other in kind of 3D space, so such as the nuclear lamina and the nuclear pore. But as I'll show you, the transcriptomes are completely different, as shown by APEX-Seq. And so coming back to one of the questions, the technique can be used to look at many different types of RNAs, no RNAs, non-coding RNAs, generally non-coding RNAs. But for the purpose of this initial study, we focused on PolyA-related transcripts, so transcripts that have a PolyA tail, which includes mRNAs, as well as many long non-coding RNAs, which are polyundated. And so here is showing the genome track genome track for one RNA, which is a non-coding RNA called XIST. And this shows you how I'm going to present the data for the purpose of this talk. So this is carrying out APEX-Seq using at different locations. And this is showing the pileup of sequencing reads. So because this data is polyundated, you can see that the reads align to the exons, but not the intervening introns, which have been spliced out. And you can see that here XIST is based on APEX-Seq and based on what we also know, it's a nuclear localized long non-coding RNA. It's actually involved in X chromosome inactivation. And as part of its function, it's previously known to go to the nuclear lamina, which is what we see. It localizes preferentially to the nuclear lamina, but not the adjacent nuclear pore. And of course, it's not bare in the cytosolic locations, including the ER membrane and the outer mitochondrial membrane. If you look at a different transcript, IARS2, so this is a nuclear encoded RNA that encodes a mitochondrial protein. And so the question really is where is this RNA really localized? And how does the protein get to the mitochondria? And so when we see the RNA, it actually localizes very specifically to the outer mitochondrial membrane. And so this observation, along with what we know about the biology of the mitochondria, suggests that this transcript is actually locally translated at the surface of the mitochondria to make a mitochondrial protein that gets imported into the mitochondria. I'm going to talk about this quite a bit at the end if I have time, but here's an example where just based on the localization of these transcripts, we can figure out a little bit about the regulation, and as I'll argue a little bit later, even the function of some of these transcripts. So these are, of course, two examples. Because this data is sequencing based, we have data from thousands of different transcripts, and we find over 3,000 of them show strong subcellular localization. So if we take our sequencing data and we project it into 2 dimensions, we can look at how the different locations are related or similar or different from each other. And what you see is a lot of nuclear locations, they kind of cluster together, so they're similar in their transcriptomes. Whereas other locations are different, such as the cytosol, the ER membrane, and the outer mitochondrial membrane. In general, from the locations interrogated, most of the transcripts localize to 1 or 2 of the different locations. And when we look at this data on a heat map, where we look at individual transcripts, so we're looking at transcripts from over 3,000 genes, what we can see is we can see patterns of localization. So individual transcripts are shown along the y-axis and locations are shown along the x-axis. And you can see a lot of transcripts are shared by the nuclear locations, which is shown in red, as well as separately by the ER membrane and the outer mitochondrial membrane. So this is looking at localization preference that goes beyond just slight localization preference for nucleus or cytosol. This is really looking at strong subcellular localization. So the fact that the ER membrane and the outer mitochondrial membrane share a lot of transcripts is perhaps not surprising, because actually within cells, they're actually right next to each other, and there are proteins that bridge them and hold them together. But what I'll show you towards the end is that there are important differences in their transcriptomes. So for example, transcripts that code for mitochondrial proteins exclusively go to the mitochondria, but not the ER, or almost exclusively. So looking more generally, what we see is when we look at the overlap between different transcripts, we can see a separation between the nuclear locations, such as the nucleus, nucleolus, and nuclear lamina, and the cytosolic locations, including the ER membrane and the outer mitochondrial membrane. Interestingly, the nuclear pore kind of seem to be intermediate between the nucleus and the cytosol. So this is the nuclear pore, of course, is a gateway between the nucleus and the cytosol, and that seems to be reflected in the transcriptome data.
So I have a quick question regarding the over 3,000 RNAs that you identified. Are these 3,000 different genes, or are some of these different isoforms of the same genes?
So these are different genes. If you start looking at the isoform level differences, there are many, many more differences. So that's just looking at the gene level.
Okay, thank you.
Yeah. Because the data, of course, is sequencing based, we can look at sequencing-wide information as well as information for individual transcripts. So when we look at data for many of the non-coding RNAs, what we find is that they're predominantly localizing within the nucleus, which is what we expect based on previous studies. So these include well-studied and well-characterized long non-coding RNAs such as NEAT1, XIST, MALAT1, and hot air. But there are some interesting exceptions. So we find, for example, some antisense transcripts will localize at other locations, including PINK1 antisense, which is the antisense transcript of a protein PINK1 that's important for mitochondrial function. So we're still working on validating some of those. But these are cases where, based on the sequencing data, you can kind of nominate hypotheses for how different transcripts might be functioning and regulating other transcripts. When we look at the location of messenger RNAs, what we find is that they can have high concentrations or high presence in either nuclear locations or cytosolic locations. So although messenger RNAs do need to go to the cytosol to be translated, what we find is that a lot of these messenger RNAs spend most of their time or most of their abundance might be in other locations, including the nucleus. And I'm going to talk a little bit about that in more detail.
Okay, so I have a question. So these non-coding RNAs that you identify here, they were picked up in the PolyA selected fraction, right?
Yeah, because they're polyadlated.
Okay, right. Did you do a similar experiment with total RNA?
I have done since. It's not in the paper.
Okay.
I don't have a slide here, but yeah. So of course you recover more long non-coding RNAs.
Right,
Yeah.
right. So okay, so maybe we'll get back to this at the end. I have a question from Rory Johnson asking how many long non-coding RNAs did you study and what fraction were detected and what fraction have a specific location?
There were, of course, fewer long non-coding RNAs because we did a PolyA selection.
Right.
I think there were a few hundred or a little over 100. I don't remember the exact number. Of course, if you wanted to really study it carefully, you would do total RNA.
Right, right. So the experiment wasn't necessarily directed at that specific purpose, but you're still able to see some of them and show that they do have specific localization for some of them.
Yeah, and again, many of them are polyadenylated, so you can pick those up. Yeah.
Thank you.
Okay, so as kind of was mentioned before, because the data is also sequencing based, of course, we can look at information at the gene level, but within the gene, we can also look at differences within transcripts and looking at some of the isoform dynamics. And so one of the things we found kind of immediately is that for hundreds of different transcripts that we recover, they have retained introns, which is something that other groups have also looked at. So this is where for RNAs such as this protein coding RNA FUS, you can see that also, although this is PolyA selected, and so most of the introns have been spliced out, this particular transcript actually has 2 retained introns that are present typically only in the nuclear locations, but not in the cytosolic locations. So here's an example of another RNA, DDX17. So it's a protein coding gene, codes for a helicase. And you can see it also has a retained intron that's present very specifically in the nuclear locations, but not in the adjacent nucleopore and not in the cytosol. And so this is something that's been studied by other groups, including Phil Sharp's group from MIT. They're called detained introns, and they're thought to essentially prevent transcripts from exiting the nucleus. So they only exit when they're ready to be used and translated. And so again, we find many hundreds of transcripts with these detained introns. In general, we find hundreds of different examples of transcripts that have alternative isoforms, so alternative 5' splice sites, 3' splice sites. We haven't really carefully gone through them or validated them, but there are hundreds of examples of transcripts that might localize to one specific place within the cell, at least the ones that we interrogated. And in general, the phenomena of RNA isoform switching is quite common. So this is where at the gene level, there might not be strong localization differences. And so that might not be reflected in the earlier data that I showed you. So for example, you might have transcripts that are localizing both to the nucleus and the cytosol, and they have similar expression levels in those regions or similar abundances. But when you look at the individual isoforms that are localizing, there might be one isoform in the nucleus and a different isoform in the cytosol. And so here's a transcript NCBP3, where you can see it has a long isoform that's only present in the nuclear location. There are other examples of transcripts that have extra sequences in the coding region or different sequences in the UTR, so the untranslated regions. And of course, for protein coding transcript, it needs to actually go to the cytosol to be translated. And so one thing that's kind of interesting is that, of course, in these cells, if the transcript doesn't go to the cytosol, then it's not translated. But in different diseases, actually, many of them can also affect splicing pathways. And some of these transcripts that normally don't go to the cytosol do end up showing up in the cytosol, where they would be translated to make proteins. So these proteins can have epitopes that are not normally present in normal cells. They can present therapeutic opportunities. They can have unappreciated or unanticipated functions. So this is one way to really explore that space. And one of the other things that's, of course, really exciting is because we have these high resolution maps of RNA localization, we can compare it to existing data out there. So for example, for protein subcellular localization, and of course, subcellular proteomics is far more mature at this stage. So we know a lot more about where proteins localize. So when we see overlapping our data with protein subcellular localization database, what we can see is the strongest co-localization is actually present at the mitochondria. So a lot of proteins that are present in the mitochondria, the transcripts localize very specifically to the outer mitochondrial membrane. And this is the phenomenon of local translation that I'm going to talk about in more detail. What's interesting is that we see also co-localization across other locations. So for example, a lot of the transcripts that code for nuclear proteins, the transcripts also spend a lot of time in the nucleus. And so this is really interesting because, of course, these messenger RNAs, they need to go to the cytosol to be translated. And so it'd be a little surprising why they're present in high concentrations in the nucleus. And so this is something that other groups have studied, where it's thought that the reason the cell retains a lot of its transcripts in the nucleus is it's a way of filtering out transcriptional noise. So transcription can be bursty, and by retaining transcripts and having a constant export rate out of the nucleus, it's a way of suppressing some of the noise associated with transcription and preventing that from leaking into translation. And so prediction might then be that transcripts whose levels really need to be carefully controlled are ones who are preferentially retained within the nucleus. And that's indeed what we found, is that transcripts coding for example, nucleic acid binding proteins, transcription factors, splicing factors, are ones whose transcripts are preferentially retained within the nucleus. Again, I should emphasize these transcripts do need to go to the cytosol at some part of their life cycle to actually be translated to make proteins. So this is something interesting we saw. If we also look at RNA localization and the underlying genome architecture, so this is focusing on locations within the nucleus. What we saw is that of the different locations interrogated, the nuclear lamina actually has the genes that we recovered the nuclear lamina have the lowest expression level in unperturbed cells, which is really interesting because the nuclear lamina and the periphery of the nucleus is generally associated with transcriptional repression and repression of different proteins are recruited to the periphery of the nucleus to repress these transcripts. And so perhaps then it's not that surprising that when we do see expression, it's lower in expression at the nuclear lamina. So consistent with this idea of the lamina being associated with transcriptional repression, when we look for the presence of retrotransposable elements, which are sequence elements derived from viruses that are present in the human transcriptome, what we see is that those elements are highly enriched at the nuclear lamina. So previous groups have shown that these RNAs that contain such elements, such as sign and line elements, are preferentially retained within the nucleus. What we find is that they really go very specifically to the nuclear lamina. And we have some other data in the paper showing the lamina transcriptome is, for example, enriched for genes that are known to be present in the DNA lamina associated domains. And the nucleolus is enriched for transcripts in the nucleolus associated domains. And so in general, there seems to be a connection between where RNAs localize within the nucleus. And this is again looking at polyundated mature transcripts and the underlying genome architecture. So this is kind of an open area to investigate further, but this was really a striking observation. So I'm going to stop briefly here, take any questions, and then go to the last part of the talk.
Yeah, I think we can move on.
Okay.
We have more questions for the end of the talk, but for now, I think we can
okay.
move on.
Let me go back. So the third part is kind of fun. Okay, so we really wanted to use APEX-Seq to understand some of the principles or reasons why transcripts localize, and so we focused on the outer mitochondrial membrane and the mitochondria in general. So this is something, the mitochondria is an important organelle. It's known to contain over 1,100 different proteins, and almost all of them are encoded by the nucleus. The mitochondria only encodes a very small fraction, so 13 mRNAs and 2 ribosomal RNAs. Most of the other ones come from the nucleus. The question is, how does that regulation really work? How does the protein or how does the transcript get to the mitochondria? And so to really investigate that problem, we suspected, of course, understanding translation would be important. And so we used translational inhibitors where we inhibited translation and saw what effect that had on RNA localization using APEX-Seq. And the two inhibitors we used are cycloheximide and puromycin, which both inhibit the ribosome, but they do so with different mechanisms. So cycloheximide actually stabilizes the interaction of the ribosome with the RNA and the nascent peptide that it's making, whereas puromycin disrupts that interaction. So they work via different mechanisms. So when you stabilize that interaction and you perform the APEX-Seq experiment, what you see is a dramatic increase in RNA localization to the outer mitochondrial membrane, particularly for transcripts that code for mitochondrial proteins. So here's an example of a mitochondrial protein, and you can see that it localizes under normal conditions to the mitochondria at the outer surface of the mitochondria. And when we add cycloheximide, it increases its localization quite dramatically. Furthermore, when we look at the transcripts that increase their localization dramatically, they're ones that are known to have high protein N-terminal targeting value, suggesting there are protein factors that actually bind to the nascent peptide and recruit it to the mitochondria. So to kind of summarize this part, let me just say that for many of the transcripts, the way they end up at the surface of the mitochondria is they start the process of translation somewhere else. As soon as they make part of the protein, the nascent peptide, the entire complex, the ribosome complex, comprising of the peptide, the RNA, and the ribosome, is brought to the surface of the mitochondria, where the process of translation is completed to make a protein that gets imported into the mitochondria. So this is kind of peptide-guided localization. This is true for many of the transcripts. However, that's not the only mechanism. For other transcripts that we find in the at the mitochondria or for other mitochondrial proteins, they don't seem to depend on this peptide-guided localization. So here's an example of a mitochondrial protein which does not increase its localization when you add cycloheximide. So it's not peptide-guided. In fact, when you disrupt that interaction that the ribosome has with the nascent peptide and the RNA, you still retain localization when you add puromycin. And in fact, you increase its localization. And so this suggests that there's another mechanism to get to the mitochondria, one that does not depend on ribosomes. And so we call this an RNA-dependent mechanism or ribosome-dependent mechanism. And so here's an example of another transcript that is resistant to puromycin. So it still localizes in the presence of puromycin. This is, again, this RNA-dependent pathway, at least that's what we call it. And what's kind of interesting is that when we look for what pathways in the mitochondria use this mechanism, they're enriched for oxidative phosphorylation and mitoribosome pathways. So these are core pathways that the mitochondria absolutely needs to function. And so there are at least two different mechanisms to get to the mitochondria, a ribosome-dependent and RNA-dependent mechanism. And it's really interesting to think in terms of really how the mitochondria coordinates its activities relative to the nucleus. So one thing that I thought, yeah.
I have a few questions here. Ioannis Tsakakis asks, how do you know that the whole ribosomal assembly goes to the surface instead of just the peptide as soon as it is released?
I'm not sure I fully understand the question. So, so it is generally thought that RNAs, at least messenger RNAs, are not directly imported into the mitochondria. So it's the protein that's imported. And so the question is, how does the protein really get to the mitochondria and get imported? And so what I'm suggesting is that either in the RNA-dependent mechanism, the RNA directly goes there and it makes the protein, and then the protein gets imported, or in the ribosome-dependent mechanism, the ribosome starts the process somewhere else, then the entire ribosome is recruited and there are proteins associated with that, and then the corresponding mitochondrial protein is made and that gets imported. So again, right. I'm not sure I answered the question.
I think we will follow up on this question because it brings many other questions in terms of why the cells would do that and how they can do that. But we'll get to that at the end of your presentation.
Okay, yeah. Let me go through just the last few slides so just I can kind of give a sense and then. Okay, so one thing that's kind of interesting is that we can also go just from RNA localization data to predict what of those transcripts might code for mitochondrial proteins. So because a lot of the transcripts, they actually increase their localization when you add cycloheximide and decrease their localization when you add puromycin, These are, again, the peptide-dependent transcripts. What we see is when we look at those transcripts more generally, we find almost all of the transcripts that show that pattern are known to be mitochondrial proteins, over 90% of them. So we asked really, can this really predict mitochondrial proteins, or at least some fraction of them? And the question is, why is that percent not 100? So what are the transcripts that show this pattern but are not known to be mitochondrial proteins? So it turns out many of them are pseudogenes, but there's also some examples. So here's a protein EXD2 that was not known to be a mitochondrial protein, but you can see it increases localization when you add cycloheximide, but not puromycin. So we predicted it would be a mitochondrial protein. And as we were putting this story together, another group actually showed that this particular protein, which is a ribonuclease, actually localizes to the mitochondria. And that's something we can see really in our sequencing data. And so I'm going to really rush through this part, but there are these two different localization mechanisms. Here's another example. And the question, what are the sequence determinants? How does the cell really know which mechanism to use? And so we developed a kind of simple random forest classifier. I won't have time to really go into the details. It's in the paper. But the idea is that when we use this classifier, we can distinguish these two different types of transcripts. So the ribosome-dependent versus the RNA-dependent transcript, which is actually shown here. And the sequences that were important were present in the 3' UTR, so the untranslated region. And they included GU-rich sequences as well as the PolyA signal sequence. So we thought the PolyA signal sequence might be a determinant, a way to separate these two different types of transcripts. And so we looked at the PolyA, the predicted polyethylation strength of these transcripts, and indeed there was a difference between the RNA-dependent and the ribosome-dependent mechanisms. We also intersected our data with a PolyA tail length data set, and indeed found that the RNA-dependent transcripts have a shorter PolyA tail length. And because there's a correlation between PolyA tail length and 3' UTR length, also looked at the 3' UTR length, and indeed the RNA-dependent transcripts have a shorter 3' UTR length. So we still don't know how PolyA signal correlates or controls localization, but that's one feature we find that's different between these two different pathways. And kind of let me just end on this last part really quickly, because I think this is really cool, is really understanding the role of the cytoskeleton in RNA localization to the mitochondria. So we predicted the cytoskeleton would be important. And so used a inhibitor called, nocodazole, which depolymerizes microtubules, which are part of the cytoskeleton, to see what effect that had on localization to the mitochondria, and confirmed by imaging that the tag was not affected. But when you do this experiment, you see a dramatic loss of localization. And indeed, the nocodazole experiment kind of mimicked the effects of puromycin in disrupting localization of a lot of transcripts to the mitochondria. And so to investigate this further, performed a time course experiment where I did nocodazole for varying durations of time, followed by performing the APEX-Seq experiment, which is just a 1-minute labeling, to see how varying durations of adding nocodazole influence influence the readout, essentially. And what's really cool is that when you add nocodazole, even for 3 minutes, you can see a loss of localization at the outer mitochondrial membrane. So shown here is the enrichment of transcripts at the outer mitochondrial membrane. And so you can see a progressive loss as you add nocodazole for increasing durations of time. And I thought this was really cool, these experiments that I performed, because you can actually use it to also figure out how quickly transcripts are lost. And so on the time scale of minutes, transcripts are lost at the mitochondria. And so this is really an example and a way of getting spatial transcriptomic information at a minute time resolution. And so this is really one observation, one approach that makes it quite different from some of the existing techniques out there. And so to kind of just summarize so I can really take questions is that, this is the graphical abstract where we have this new technique and we use it to get insights into mitochondrial and nuclear biology. And before I take questions, let me just acknowledge both my advisor, Howard, as well as Alice and Shuo, who contributed to this work. And Shuo is an equal contributor to this work, really, as well as acknowledging some other people whose work I haven't had time to really talk about, kind of continuing this work, as well as funding sources, the K99 and the Beckman postdoctoral fellowship. So thank you very much, and I'll take questions, yeah.
Okay, thank you very much, Furqan. That was very well done. It was very interesting. We still have quite a few questions that we can take and answer. I've saved some during the presentation, and we have time for those. Let's start with, I had different people asking whether you tried using APEX-Seq on centrosomes or the Golgi.
Haven't tried it yet. Well, so part of it is, of course, we made this technique, and my hope is that it will be widely used. So we have a protocol, but if you want to try it, please do. Yeah.
Yeah, is there any specific location to targeting specific cell compartments? Is it just finding the right apex construct that will allow you to have a very specific localization, or are there other factors that have to be taken into account when you design these experiments?
Yeah, so finding a good protein tag, of course, is very important, and confirming by imaging that the tag does indeed localize. So of course, at this point, there have been a lot of studies using apex proteomics. And so pretty much any of those constructs that have been used for proteomics can be used for transcriptomics as well. It's the same construct, and you can also get proteomics as well as transcriptomic data from the same construct. So do them side by side. And so, yeah, that goes into the study and the design of the experiment. But yeah.
Okay, we have Yuri Bobinik asking, with respect to limitations, have you looked at other cell types? They said that they've had problems using protein labeling with APEX in liver cells due to high intrinsic biotinylation of proteins there. So do you think this could interfere with RNA labeling in this or other systems?
Yeah, so I've done experiments also in a different cell line in collaboration with other groups, breast cancer cell lines. So, but of course, if there can be differences, and if there is, yeah, high levels of biotin, there could be particular cell lines where it doesn't work. That's what I've heard also anecdotally. Yeah, I don't have much experience looking at many different cell types.
Okay. What about, okay, I have this question about whether you've tried using split apex as an approach, and whether this could improve signal to noise ratio. I'm not personally familiar with split apex. Can you tell us a bit about it, and whether you've tried it?
Yeah, so split apex is also developed by Alice's group, where you split the apex into two different parts and you attach it to two different proteins. And the idea is that they'll come together within the cell and get you information about, for example, contacts between different organelles. So the one that would be interesting that Alice's group has explored before is looking at, for example, regions where the outer mitochondrial membrane and the ER membrane make contact with each other. And so the split apex paper, I think, was last year, and we haven't really tried it. I think that the signal to noise wasn't really very good in the split apex approach. And again, the paper is there, but it could be. I mean, it's a challenge getting it working. So it's an engineering challenge that Alice's group has, of course, worked on, into really getting it working so that it only associates in cells when both partners are present. So it is a challenge. I haven't tried it yet. I don't think the current version would work really well. But of course, that's an ongoing process. Yeah.
Okay. I have two questions from Kevin Tsai and Alex Price that kind of overlap. So they ask, if the labeling introduces an RT stub, does that mean that your library prep has to use circular ligase? And would that introduce a bias in your data set? And the question is, can you remove the biotin tag from the RNA before library prep?
Yeah, so I'm using superscript 3, which is what we use in the initial paper. So that actually stops when there's a modification. And so that could introduce a bias, but in practice, it does not. And the reason for that is that the labeling doesn't happen very frequently on the transcript. And the way we do is that we actually, I can talk about this in more detail, but essentially we pull down the transcript and then we fragment it and sequence. And so most of the sequencing reads come from regions that were not biotinylated. So it turns out not to be a problem. Of course, different modifications, different RTs might read through them, and that would get you to also look at other things, long read sequences and different iterations. So that's an exciting area to explore.
Right. Okay. Clemens Ertel asks a very interesting question, and I think this is something that we were about to get to. It's almost a philosophical question. So Clemens asks, if the peptide guides the RNA away from translation initiation factors to the site where the protein is supposed to be, then it is anticipated that only one or very few proteins are made from that RNA molecule, and it seems to be biologically expensive. And I had the similar kind of questioning with regards to why would the cells transport the RNA to the site where the protein needs to be? Is it because energetically speaking, it's just faster to and easier to move the RNA, but then you're stuck with the problem that Clemens suggests here? So have you thought about this, and do you have any idea of why that would be? I'm thinking of known examples where this happens, in particular with local synaptic translation in neurons, where it's pretty obvious that if you have processes that happen in a time scale of milliseconds in cells that are several millimeters or even centimeters long, you kind of need to have local translation going on. Now, this is a phenomenon that you're observing in hex cells. And so the question is why? Why would that happen?
Yeah, so that's a great question. There are actually two questions within that. And so one of them we contribute more to. So the first part is, why is there local translation? And so an example, for example, in neuronal cells where there are long projections and the distances are large, it would make sense to transport the RNA long distances and then locally translate it to make proteins. And that's something that people generally appreciate at this point. And so we still don't understand really in a small cell like a hex cell and all, why is RNA localization so widespread, and why does it depend on all of these different, like the cytoskeleton and all? So that's one question. The second question is specifically, so what was thought to happen previously, and the paradigm in the field is that transcripts are repressed from translation until they get to their particular location, and then they're exposed. Often they might be ubiquitinated or modified in some way, and so then the transcript actually gets translated. What we see is that for these transcripts that go to the mitochondria, they start the process of translation and then they're moved. And so then the question is, would that be problematic? And so that's something that we've observed. That's not the common paradigm, but there are some imaging studies from a couple of years ago that also suggest that you can start the process of translation and then move the ribosome complex before it's finished translation to a particular location. And that's something we see in our data set. And so why that would be advantageous is something that, it would be interesting to speculate. Of course, at the location, there are ribosomes as well. So the mitochondria is decorated with ribosomes, and some of the factors, maybe they can go across them. So there's be a high concentration, but that's still a good question. And that's something that I would say we've contributed to in really showing that and supporting that. And, that leads to more questions of really why that happens. So I don't have a good answer, but I'll just kind of distinguish between those two different phenomena.
Great. Simone Eber asks, have you ever directly compared APEX to BioID?
No, I have not. I can't compare that. So people have looked at proteomics for that, though there haven't been that many good direct comparisons, but I haven't done transcriptomic work with BioID.
Okay. When you looked at long non-coding RNAs, did you use known annotated long non-coding RNAs, or did you also do de novo detection?
There's no de novo detection. So this is all aligned to the ensemble transcriptome. So not RevSeq, but the other one, the ensemble. So there's about 60,000 different genes. But yeah, I mean, you could do de novo as well. Yeah, that's not something we did.
Maybe one or two more questions. Okay, I have this question from Ioannis Tsagakis, who says, assuming you could target a protein that is found in most liquid droplets in the cytosol, do you think APEX could be used to find out the proteome of phase separation liquid droplets? So this is probably more on the protein side of APEX.
Yes, you can. I think there's actually already a couple papers on that from a year or two from a group at Berkeley. I forget their name, but they've actually, I think they've already looked at that using APEX proteomics. So yeah, it can be done. And I should also say that, yeah, you can look at phase separated condensates in general using APEX.
Right. Okay. Razin Barutku asks whether you've ever directly compared APEX-Seq to CLIP-Seq. It's kind of funny because, you know, APEX-Seq is kind of a better version of APEX-RIP that's used to be performed before. So can you tell us about direct comparisons between APEX-Seq and CLIP-Seq?
Yeah, so of course with CLIP-Seq, you get some of the direct interactors. And at this point, we're not looking, so because this is proximity based, and so you wouldn't get the kind of high resolution map that you would get with a direct interactor. But of course, in using CLIP, you're only looking at direct interactions, and sometimes you're interested in indirect interactions. And you depend a lot also on how well the antibodies work. And of course, there are a lot of different controversies where the antibodies don't work, and this is particularly true, of course, on modifications where there's so many different. So it's a different thing, and I think that they both together will make a better story and a better really way to look at that. Yeah, but this is of course looking at interactions that are close by, which can include direct interactions. So it's a little different from CLIP, but CLIP, of course, you can, at least in the way APEX-Seq is done here, you are losing some of the really resolution within a transcript, but that's something that you can actually try to recover to some extent using a different approach. Yeah.
Okay. Now we have a question regarding the time frame for the labeling. You use a 1 minute pulse. The question is, if you were to use a shorter pulse, would you be able to look at faster kinetics? And there's also something that you didn't really mention today, but that's in the paper about how when you're looking at the transcripts that are labeled at the nuclear pore, it seems that transcripts that just quickly go through the pore, you're not labeling them. So would you have to use different methods to get to this kind of labeling, or would using a shorter pulse get to that issue?
Yeah, so you could use 30 seconds. You could probably even do 20 seconds. So if that's that would help with the way the experiment is designed, that would definitely work. I wouldn't go much longer than a minute. I don't think that's necessary. If you do much longer than a minute or a few minutes, you can lose some of the good localization data, but you can go shorter. So the question, then the second part is, of course, this is not a single cell approach, and it would take a while to get it to somewhere close to that. So you're looking really at an ensemble average information from typically you can do this in this iteration for 1 million cells. You could definitely go a lot lower than that, but this is an average information. And so if a transcript is not spending much time at a particular location, then you don't pick it up. And that might explain what you mentioned about the nuclear pore.
Okay, great.
That it's just, if it's just passing through really quickly, then it's not spending most of its time there. And so it might be challenging to pick it up at that location.
Okay, so let's take one last question. This one is a long one, so hopefully I can get this right and we can understand what the question is. So no-code-azole disrupts ER dynamics, and the mitochondria directly interact with the ER via ER mitochondria contact sites, which raises the question of the biological specificity of the no-code-azole data regarding the localization of the mRNAs at the mitochondria. Can you exclude that the interpretation that the disruption of mitochondrial RNA localization after no-code-azole is pleiotropic consequence of just disrupting the ER microtubule dynamics?
Yeah, so that's a very good question. So of course, because the cytoskeleton is so important and it's plugged into many different processes, the mitochondria itself moves on different cytoskeleton pathways. How can you assure or confirm really that no-code-azole is indeed doing what you think it's doing? So what I'll say is that if you do the no-code-azole treatment for a long duration of time, like more than 30 minutes, you definitely see an effect on the mitochondria in terms of where they're localizing, and the cell kind of looks different. when we do this experiment for a couple of minutes, you can see our RNA localization loss, but almost no really change in where the mitochondria is localized. And that's really the fast time course information that I showed you. And this is, again, kind of going back to this particular slide. So even in a time scale of 3 minutes, you see a dramatic change in localization. To kind of say some more, since then, I haven't shown data, but also trying to understand some of the specific motor proteins that might be attributed. So instead of using no-codisol, which is kind of not a great way to look at this, to actually ask which is a specific motor protein, we've implicated other motors that specifically walk on the microtubules and actually are involved in this process. So I have other data that I haven't shown you to suggest that this is indeed what we're looking at least at these short time points, like 10 minutes, 15 minutes. So yeah, but that's a good question. That's definitely something to think about, and definitely it is a concern.
Great. Well, thank you very much, Furqan, for your time. Thanks to all of our attendees. Again, please join us for next month's Journal Club. I want to mention that we've had over 100 attendees throughout the talk. So thank you very much for being here and making this a lively conversation. It was a very fun Journal Club, I think very informative. So we hope to see you again next month. And thank you again, Furqan.
Yeah, thank you so much for having me. I'm so glad so many people could join. And, you know, it's a difficult time, but it's fun to kind of be here and have people really participate. Yeah.
Absolutely. I think it's a great way to keep the science alive.
Okay. Yeah, again, thank you for having me.
All right. Thank you all.